Okay, and just before introducing Vânia to all of you, I would like just to once more, for those that are not yet familiar or that may have heard just a few words about what is MPS at Nova and why are we here today. So I would like just to say very briefly that MPS at Nova is a training led by Sarela here also today and Claudia. Santos. So it is a training project led both by ITKB Nova and Nova Medical School, two organic units from Nova University of Lisbon that aims to give extra resources and technology to both these the researchers from these two institutes in Nova University of Lisbon, mainly into MPS systems, so microphysiological systems, the conventional and perhaps more known for all of you of organ-on-chip technologies, organoids, and even stem cell technologies. So mostly to study chronic disease and mechanisms of host microbe interactions. Very briefly, this MPS at Nova training aims to, as I said, develop and bring this new technology of microphysiological systems to ITKB and to NMS, mainly by training and through research fundings. And we have some core areas of expertise that were put in the grant agreement, mainly towards brain, skin, lungs, gut, of course, liver, and even eye and retina, but others have been joining, and we will try to develop our technology and our knowledge and hands-on proficiency into these new and advanced technologies of microphysiological systems, both regarding host microbe interactions and the mechanisms of chronic diseases. So this training will capacitate the researchers from these both institutions, as I said, through training and through funding the research. And just to remind all of you that there are open calls already from this training for all the members of MPS Nova, namely some exploratory research projects. It's a small seed funding for researchers to start working into these microphysiological systems technologies. These calls are open until January 15, okay? There are also some staff exchanges, and this perhaps will be the core of this training. So to give hands-on expertise to researchers from Lisbon that go abroad to learn technologies. For the moment, there's a staff exchange open for 2 researchers from our training to go to Milan, to Technopole, to learn more about reprogramming of human cells into stem cells, and even how to manipulate stem cells and iPSCs. These staff exchange in particular will be open not only this year, but also the 2 other years of this training. And of course, for the time being, there's also resources for publication fees called also open. So, and this will be on a first come, first served basis. Perhaps now it's still very early, but keep in mind there is these funds to support open access publication fees in the field of MPS, and also some conference fellowships for researchers from either RTKB or from NMS to go to international conferences in the field of MPS abroad with the support of the training. Okay, and this was just what I had to say before introducing Vânia. So I would like to say to begin with thank you so much to Vânia once more for accepting our invitation here to be the first speaker of this virtual seminar series. This virtual seminar series, for those that do not know, will happen every 2 to 3 months online in the same format. And all of you will receive information in advance, both through social media and through the communication that Paula, our project manager, has been dealing with so well. So keep attentive to this, and we'll have more upcoming seminar series following. But first of all, I would like to introduce Vânia. Vânia is a senior researcher at INESC, leading the Microfluidics lab, and her work has been focusing on technology and developing microfluidic platforms and organ-on-chip systems. Vânia holds a PhD in mechanical engineering and a degree in technology chemistry from University of Lisbon. She, for the moment, serves as a convener of different ISOs, and microfluidic standardization, and she's also an expert in other ISOs and from CENELEC on organ-on-chip standardization, and she will talk us more about these standardization and these regulations on organ-on-chip technologies today. And besides that, she's an invited assistant professor at Instituto Superior Técnico, also here in Lisbon, where she teaches microfluidics and supervises PhD, masters, and bachelor theses. And besides her track record, I would like to say that perhaps Vânia was one of the first leaders here in Portugal working on MPS, on microphysiological systems. So I think all of us will owe so much to her. And thank you so much, Vânia, for accepting to be our first guest here today at the virtual seminar series. And thank you so much.
The internet is saying that it's a bit unstable and I missed the last part, but this means I can start, right?
Yes, yes.
Okay, great. Thank you so much for the introduction. It's a huge pleasure to be giving this seminar, and it's really nice to see that indeed when people connect, things happen. So I met some of you at EuroOx in Milan this year. We had the pleasure we didn't know each other. We had the pleasure to talk a bit and discuss a bit about this standardization and the necessities and all of that in some of the sessions, and also with some of you. So it's really nice to see that it's going and it's getting to places, and people are more and more interested in getting things done in a very structured way since the beginning. So it's really, really nice. So I'm gonna start my presentation. I don't know am I sharing my screen to you? Because it's not showing here, so I'm not sure.
Yes, it's perfectly.
Okay, perfect. Because I think it stopped. Okay. And you can see my laser pointer, right? Yes?
Yes.
No? Yeah? Okay.
Yes,
So it's everything is running smoothly. So part of you probably already know what an organ on a chip is, but I wanted to focus on something. So we have in organs on chip several components. We usually had the tests in vitro, meaning that we had equipment in the lab to test the animal, to test the experiments. Then we have the models in vivo, that means the animals, and organs on chip is just a combination of the two. So putting cells into something that is in the lab that is not an animal. And this has been gaining some traction, actually a lot of traction. Usually these devices, they are up to now very simple ones with channels and chambers. They have cell cultures. Now they are starting to gain some sensorization and actuation to actually understand what's happening within the cell tissue. And of course, they can be used for several things like testing drugs or disease modeling or personalized medicine. So these organs on chip are actually being used very efficiently to have better results, more precise results, and results that may be used directly in humans, which is not the case when we use animal models. And this has been pushed mainly because both the FDA and the European Commission are making a big effort in reducing animal testing. So the 3 R's, the reduction, and the other 2 R's that I never remember when I need. Of course, if we can test human cells directly, we can enhance the accuracy. And because it's a small number of cells and we don't have to have animals and take care of the animals and reproduce the animals and all of that. So it's the costs associated to this development and testing is a lot less. One thing that I've been noticing is that indeed the microfluidics realm has had an expansion sooner, because microfluidics is used, and that is where I work the most, is used in many different applications. Not only in the biology, but chemistry, physics, mechanical engineering, civil engineering. So you name it, microfluidics can be used there. So it tracked it a bit sooner, and then organs on chip are now, since '83, they are being discovered, tested, enhanced. And we are starting to see a lot of discussion on this subject, which is quite positive. I need to take you out of here because you are in front of my screen. For organ on chip, there are 4 main trends, and apart from integration of, for example, sensors and actuators together with the cells, and the applications mostly in point of care and personalized medicine. And of course, coming a lot from the international collaboration, we are seeing the interest in the development of international standards, because mainly we need to compare results, and we need to understand exactly how the results are being obtained, and if they can bring information that is comparable and has satisfactory information to understand whatever it is that is happening to the cells. And we want this because we want to bring the manufacturing of these types of devices at the same level of maturity and industrialization as the electronic devices. Basically, I want my organ on a chip to be able to communicate with this equipment from this company, that equipment from that company, that tubing from that company, those connectors from the other company. And I don't want to even think of that. I just want to focus on my study inside my organ on chip. And I don't want to take care of all the engineering that is behind the functioning of my organ on chip. It's more or less, as I am a user of my computer, I don't want to take care of putting the electronic components together to make the computer work. Basically, is something like that. So for this standardization in microphysiological systems, and in particular organ on chip, we are trying to gather people from several realms, from industry, from academia, from regulators, to put the standards together, and we will see what exactly is a standard in the next slide, to be able to characterize, compare, and allow a clear communication that it is understandable by everyone. So here the standards are developed as a result of the work of different collaborating stakeholders that we see here, and they will help to do all of this, and with the assurance that indeed the results are comparable. So if we are sure of what we are doing, then the technology will go forward a lot faster. Before going to see what a standard is, I'm just gonna show you this. So this week I had to see some things related to polystyrene, one of the materials that we use to develop these devices. And I came up with the technical characteristics that have the density, the mechanical properties, optical properties, thermal properties. And if you see here, all of those are measured with standards. And then they have a value and a unit. And because they are measured with a standard, I know exactly how they were measured. I know I can compare with the results of other suppliers, and I can better decide what material, if I will have the material from this supplier or another supplier. Actually, if you have had the pleasure of developing microphysiological systems, you know that the material is extremely important to the successful growth of cells. So it's very important to actually understand the specifications of the materials. And because they are specified using standards, then I am sure that the values that I have for each of the units, the variables, is the accurate one. And it has an error and an uncertainty associated with it, and then I can work better with that. But then if I have lack of standards, this will negatively affect the reproducibility and comparability of the results. So it's very, very difficult that way to promote the active use of these devices. More important than that is very, very difficult to guarantee interoperability. The thing that I was saying before that, well, I can use the device from this supplier and then several parts from other suppliers, then they don't interoperate together, and it's really, really difficult to get one device working for me. So I need to spend a longer time just to make sure things work. So what's a standard? A standard is a document, okay? And this is very important because I hear standards talking about standard this, standard that, and I'm not quite sure if people understand exactly what the standard is. It's a document, it's a paper with things written on it. It is established by consensus, so it's not just a protocol, for example, that is defined by one group. It has a lot of people involved, and you will see a bit about that later. And it is approved by a recognized body. In our case in Europe, is approved by CEN, and then goes to ISO, for example. We can actually have national standards. We also have that through IPQ. And it should be based on consolidated results. It's not just that I write a protocol, I send it to go to a standard, I did 1, 2 tests, it went fine, and that was it. No. It needs to have intercomparability between several players. And then after comparison of all the results, then we can make sure, we can be sure about the veracity of the results and the accuracy and the reproducibility. So this standardization is no more than the activity of, and this I will have to read because it's a very nice definition. It's the activity of establishing with regard to actual or potential problems, provisions for common and repeated use, aimed at the achievement of the optimum degree of order in a given context. It consists in a particular way in the formulation, addition, and implementation of standards. It's quite cryptical, but in the end, it just means the whole process of putting the standards out. Things that are very important is transparency. It cannot be proprietary. Imagine a proprietary protocol for measuring something that will not go to a standard. It needs to be seen by everyone involved, and then it can be purchased online. It needs to be open, representative, impartial. It needs to be obtained by consensus. It needs to be coherent and effective and relevant. And this is extremely important. Just to give you an idea, a timeline for a standard is about 36 to 48 months, and it has all of these steps. So it comes into a new work item proposal, and then it goes through several steps until the end, the last document, which is available online, which is an international standard. I will just leave it because you will then be able to see all the steps if you want. Also go to the links, but I will not talk more about this. Just to have an idea that the discussion is taken throughout all this process. The documents go back and forth amongst all the people involved and ISO, and they need to to have a language according to ISO standards also. So it's not that we can use words at ease. Each word has a specific meaning, and that then is checked by ISO collaborators. So how do we do this? Well, we find which standardization technical committee and working group is interesting to actually give some input. We identify if our country is participant or observer in that technical committee and working group, because if it is participant, it can vote. But if it is observer, it can give input, but cannot vote for the standard to come out. So this is very important. If you are really engaged in this technical work, to make the country, to make your country participant, so you can actually vote for the standard to come out. And then you register as an expert in your country. They will then register you in CEN if you are in Europe and in ISO internationally. You check if there are relevant standards that not only are going to be started from scratch, but are going to be revised. And then you give your comments, give your input. And of course, if discussions arise, then you need to explain exactly why you are suggesting things as you are suggesting, because in the end, the decision needs to be consensual, or else when you are voting, your decision may not pass. And then that may mean another year discussing things. So one of the things that happened, for example, in a project that ended recently, is that the consensus was really easy to reach. A lot of stakeholders were involved. We had several discussions, but we could put standards out in 2 years instead of the 4 or 5 that are usual. So it's really, really important that you have a good dialogue with the people involved. So for me, for example, when I talk about finding working groups and technical committees, one of the things that I did was to look for standardization experts and technical committees and working groups that made sense. For example, for microfluidic devices, both at ISO and at CEN. So I'm leading those 2 groups. If you are interested in the engineering side of organs on chip, I'm the person to talk to, aside from all the experts that are in these working groups. I'm also an expert in 276, which is more related to the biotechnology, so more related to the cellular part. And the focus group of organ on chip was the one that established the roadmap for organ on chip for the standardization of organs on chip. And then there's also the IEC group that relates the sensorization part, so the electronics, to the organ on chip, which is quite interesting. Just to give you a bit more information, for example, there is, from the focus group of organ on chip at CEN, now there is a new subcommittee in 276 for microphysiological systems and organs on chip. The person in charge of that, leading this group, is Massimo Mastrangeli. He's in Delft University. There's also working group 3 for TC48, technical committee 48 for the engineering part. There is working group 4 of TC276 for cells and related entities, and then this IEC that I was talking before. And as you see, we have here representative leading from the Netherlands, from Portugal, from Japan, from Canada. So we have many, many different countries involved in these discussions. That's why it's important that your country is a P member, so you can vote. Your inputs then count. So for this roadmap of standardization for organ on chip, we had, so you see, a 2-year work. So you have a QR code here that you could use to access it. We had 120 European experts, so it's not just a group of 3-4 people. 10 focus group meetings, 90 working group meetings. We had 4 working groups, 3 revisions of the whole document, and it was published in July. So we are really, really happy about this. And we looked for areas that required standardization. So we gathered the people involved in groups. We understood the areas needing standardization, and then we went and see, okay, are the standards existing or not? Is there anything outside, any standard that could partially relate to what we want to have? So we can drink from some of that information, and we don't need to start from zero. Who are the regulators that could come in and give us some advice on how to proceed? What are the most important aspects to go to standardize first? One very important thing is we never standardize whatever it is inside the device. So your design is your design. How the chip connects to the outer world, that could be standardized. The same way as a USB stick. So whatever it is that you have inside your USB stick, either the electronics or the documents, it doesn't matter. It's your thing, but you can fit it to any computer that has a USB stick or a USB-C stick. Or so more and more. So 20 years ago, we had, for example, each of the phones had their own charger. Now all the phones have except for Apple, all the phones have the same USB-C for charging. So that's going towards standardization. And in this roadmap, we have identified 70 documents, guidelines, standards that are useful for example, terms and definitions, cell and tissue sources, material specific, sensors, modular integration, data management, use of human tissue. So a lot of different documents that are already out and that could serve several of the purposes that we have identified in the roadmap, but also could leverage new standards that are necessary specifically for this technology. For example, in the engineering part, we have developed 4 standards under the MFMET, establishing metrology standards for microfluidics, for interoperability, for liquid handling, vocabulary, and specifications for pumps. Why is this important? Because I want to make sure that my device relates to others. So let's just see in more detail. And you can also find this information. One of the things that we are trying to do is always have the information before in white papers that could be spread around, that could be tested. We collect inputs, and then, only then, we go to a standard. But what does it mean? So we have our corner, and we assign an exclusion zone of 3 millimeters, so nothing happens in this area. And then we start with the ports that can be fluidic ports or optical ports, electronic ports, electrical ports, well, you name it. And they are defined in a grid. And this in particular has a pitch P that it can be a multiple of 1.5 depending on the diameter of your ports. And then you can also use this grid when you are defining your device and you want to actually fabricate that. You can specify for your consumer, well, my first port is in A1, my second port is in A3, and it also has a port in C2. So me as a user, I already know that chip has these ports, so I need to use connector X. And that is easier for me as a user to use the device. It also explains how the size matters and what sizes could be used. For example, standard sizes as a microscope slide or a double microscope, a credit card, or multiples of 15 millimeters, both in X and in Y. Another example of standardization or attempt of standardization is trying to figure out if we could specify our devices by pressure and temperature. So maximum pressure of 200 or 700 and maximum temperature of 50, 75, 100, and so on. And this comes from the study of what is being mostly used by microfluidics users, but also manufacturers of these devices. And then we have also the capillary devices that are a bit different because there's no pressure as this involved. It could be, they could have temperature, but we are still trying to define that. And of course, if we are talking about flow, then we need to measure the flow. So we need protocols that everyone can use to measure the flow. And why is this important? So this was done by the gravimetric method, which is actually having a scale. You have your fluid going through the syringe, passing your device, and then dropping on top of the scale. Of course, if this is microfluidics, means also that the flow is very slow. So evaporation is an issue at the outlets, right? And so this, that is a standard method for larger dimensions, had to be adopted for these very small flow rates. And what we see here, and this is non-dimensionalized by the nominal value for 1,000 in black, 1,000 microliters per hour flow rate, very stable, very nice. This was done with a syringe pump, with a Nexus pump that the supplier says it goes to picoliters per hour very easily with a good uncertainty. However, there is still at this point, so this was done in 2020, at this point there was still no good protocol that could evaluate this very low flow rate. And what we have seen is that with this pump at one microliter per hour, meaning 1,000 more, we had a variation of 2-fold. So what this means is if I, as a user, use a pump where the manufacturer states that it has that drives flow at a certain flow rate, but there are no methods to actually guarantee that flow rate, not guarantee, to measure that flow rate, it may mean that I introduce liquid inside my organ on chip, I program my pump to, 1 microliter per hour and expect my cells to behave in a certain way, and all of a sudden they are behaving very differently. And that may mean that the shear stress is a lot higher than what I was imposing or than what I wanted to impose. And that may hinder my results. And then I am like 6 months thinking and trying and changing and redesigning and designing and redesigning, and nothing works. And all of it would be just a matter of calibrating my pump. So for this matter, in order to calibrate pumps at very low flow rates, there were no transfer standards, meaning devices that can be used to calibrate flow meters that then will be used to calibrate pumps. So this is like a chain, a huge chain. So what we did in in another project, it's called MED2, Metrology for Drug Delivery, was to develop a transfer standard for flow rate down to 5 nanoliters per minute. And this is based in a glass slide, a microscope slide format of 75 by 25 or 76 by 26, so it's in between. It has the ports in those positions that I stated before, the exclusion zone, and then it has actuators. It's an electroosmotic pump. And we did a calibration test that ran very nicely at 84 nanoliters per minute, meaning 5 microliters per hour, with a very, very good uncertainty. So it means that, and if we see before, we had the 23% uncertainty. We could, with this device, after calibrating the flow meter, we could really reduce the uncertainty in the measurement, which is very profitable. And in relating to these standard footprints, we also developed another one with a microtiter plate footprint. So it's a standard size. This one was for 96 wells, and we introduced flow in these 96 wells. So we could study the flow, the behavior of the cells in the flow. In this case, they were to study angiogenesis, so how the nucleus of the cell would behave according to the flow. And it was really, really nice to see, and actually it was really demanding to actually get the cells behaving nicely on top of these islands. So each of these islands represents a well, and then the flow goes on just on top of the island. It doesn't go up and down. And it was required to have all the channel at the same, with the cells feeling the same shear stress. So this was engineering, in an engineering point of view, it was quite demanding, but in the end, we achieved very, very nice results. So the idea here is actually to already use the standards that are out there to define very simple strategies to mount or to design, fabricate, and mount my device in order to be a more universal device to the outside. So it can be modular, but I know other modules from other suppliers can fit my own device, which is quite good. I'm just having a picture here of the screen. Okay, done. And we can do this at INESC MN because we work in a clean room facility. So we have the capability of resolve very small structures down to 30 nanometers. In a more common lithography, we go to 1 micrometer, which is totally okay for our microfluidic devices. And we also have the possibility of integrating sensors and actuators, MEMS, and simulate all of this, all this multi-physics that comes from the materials, that comes from the fluids, that comes from the cells, the chemicals, and put it all together in one single simulation to understand a bit more on the flow. So I show you here some examples of our devices. For example, a simulation of a trapping chamber. So cells get trapped in this center, and then they have pillars around, so the fluid can go in and out both ways, both left to right and top to bottom. So this is a study of what happens to the pressure and actually vorticity and velocity, depending on where the flow is coming from. We have cell chips, we have microfluidics with MEMS, so actuation, mechanical actuation, but we can have sensors in very small needles to fit in these devices, or have platforms that include the sensing part and the actuation part, and then the fluidics part. This one is smaller than my pointer. Actually, you can see more or less the size of a nail. It's a lot smaller than that, and includes all the sensing, the actuation, the electronics, and the fluidics part. But it can be a bit bigger. So here you have an Eppendorf, a small Eppendorf, and all the electronics and the fluidics part with the chambers. And these are for detection, but we can grow cells in these devices exactly in the same way. Because it was necessary to establish these metrology standards, we had a project that ended beginning of this year for specifications, for development of measurement protocols, for guidelines for interfaces and connectivities, and then for dimensions and modularity and sensor integration. I will not go through all of this, but just to let you know that all this information is available at the website. So 4 standards, 8 deliverables, 6 short videos that are really cool on how to measure several variables, 24 reports, papers, white papers, so a lot of information that could be useful for you. It's free to look. It's open to everyone. So it's a good starting point to actually understand that there are already protocols out there that are stable protocols that were tested by a lot of different partners, and that in the meantime will go into standards. For example, the webinars, they are for the flow, the wettability and roughness, the dimensions, the interfacing, and leakage. So leakage is a big issue in these devices. And these are 10-minute videos that explain briefly how the protocol goes, which is a lot nicer than reading a 10 or 15-page document. So with all this information, we go and discuss with a lot of actors that are engaged in standardization, and we put this into standards. Actually, now, this last month, we had a new consortium for the follow-up of MFMET. So we now will have the MFMET 2 for quantities, flow, with and without particles, with and without cells. For materials, material characterization, I think I got this, it's exactly the same. So it's not exactly the same, the 2 work packages, my mistake. But it's one for material characterization and the other one, I should know this because I wrote it and I defended it. So we will define as standard device to test quantities. And this should be more of the quantities. Let me see. So for example, in work package 2, we will focus on sterilization, on wettability, on deformation of materials. Work package 3, I'm not remembering what it is, but well, it's fine. Then I will correct this if you want. And this information is important because we have already established a roadmap for the standardization in the engineering part that is also related to organs on chip, for leakage detection, for the assemblage, for wettability, sensor integration. I know one of the work packages is for a sensor and actuator integration. So they were 2 and 3 were had the same written, but one is for sensor and actuator integration, and the other one is for materials studies. Then we have the quality control and the qualification, and all of these. So you see, they take a lot of time, especially if they are standards. If they are technical specifications or international workshop agreements, they are a lot faster to achieve. But if they are standards, it may be that it takes a bit more. So the final message that I want you to take home is that we really, really need, we really need the international collaboration, because only this way we will have all the countries involved in the voting of the standards to vote favorably so the standards can come out. So it means that all the process is faster. If we have international collaboration, we also have the ability to make the protocols and the guidelines available for testing. And if they fit everyone, we can harmonize the documents faster, and we can reach a consensus faster. And most importantly, if we have international collaboration, we test faster, a lot faster. Because we know we are competitive, everyone is a bit competitive. Even if we are collaborative, we are competitive, so we make things better and faster if we are together. And then with the regulatory support, financing, and having the regulators on board to actually drive us towards the right direction, it has been very, very important. So that's what I wanted to tell you today. There is a lot of information out there. Actually, you can, for example, in the roadmap, you can see one of the annexes has all those documents that I mentioned, all those standards and guidelines. They are already here for you to see and actually understand if they make sense in what you want to do in your device, in what you want to study, and what you want to guarantee. And of course, it also has an important part, which is the recommendations, which is the work that is going to be done now in the new subcommittee for microphysiological systems. And this is to define, for example, the vocabulary for this part, or it's called the terminology and the definitions and the symbols, the minimum reporting requirements when you are showing your results. How do you measure and qualify materials? How do you control the flow and measure the flow? The microfluidic connections, for example, or how do you organize a study design or an experimental protocol, how it should be described? How do you acquire and store the data that can be used not only by you, but by the community? And more we are seeing databases, very, very good databases, but each of the database has their own way of storing the information. So at some point it's a bit difficult to compare. Or for example, applications, regulations, the use of these models, how to protect the autonomy of patients or donors, how to provide guidance to the industry or to the doctors on how to use these technologies. Okay. So you didn't make any question during the presentation, but I'm totally up to questions now.
Thank you so much, Vânia. A lot of information to dig in. But super important, and certainly the road to go. Are there any questions to Vânia? Oh, look, Claudia's there with Sandra in the office. Yay! Any questions from the audience? Please feel free to shout or raise your hand if you want.
So we have questions in the room. Go ahead.
So except those countries that you mentioned that they are part of this standardization and community, the other part of the Europe they are not, and the club. So you mentioned like not even Europe. So you mentioned Japan, Canada.
No. At ISO level, so we have several levels. So ISO is at the international level, meaning any country in the world can participate.
Okay.
CEN is the European Committee for Standardization, and that is just for European countries. Then there is the Japanese Standardization Committee, the US Standardization Committee, the Australia Standardization Committee. So there are several depending on the regions in the country, but all, so standards can be national, and you have your own standardization body. In Portugal is IPQ, Instituto Português da Qualidade. Then you can have, in Europe, a European standard at the SEN level, and then you can get this into ISO for it to be an international level.
Okay,
So a national standard has to have consensus within the country, a SEN needs to have consensus within Europe, and ISO needs to have consensus in the world.
so you can go directly to international, or you need to go this
So
step by step?
we, how we do it in the microfluidics standardization technical committees is we always submit to SEN, and SEN then sends it to ISO. Okay, so we have like the path is through Europe into an international standard. Okay? And that is important also for the metrics of each of the standardization committees that that have their own metrics, and they push their countries, their associated countries, to actually work on and lead the development of the standards.
Vânia, I have like a more general question. So imagine us as end users, how can we help these bodies to speed up the process of standardization of microphysiological systems? Of course, we are starting and we are trying to start the best way possible, but how as researchers can help in this process to make it faster? Somehow.
So do you remember during the talk I mentioned intercomparison
exactly
that we need to do to establish the protocols? For example, imagine that within your work you develop a protocol that you cannot find anything related anywhere, so you develop your own protocol. So you can reach the community to actually get the protocol to several players so they can also test. So what we do is we have a very good established community that we want to call more people in, that actually help us to have results, to be comfortable about putting the protocols out, or the guidelines on how to, I don't know, design something or mount something or fabricate something or test something.
But imagine this is applicable to all the layers, let's say, of microphysiological system, not only technological, but for instance, if my cells in the hands of other researchers give the same results, for instance, in the same microphysiological system, something like that.
Exactly. So imagine that one of the things that we are working on now has to do with the sterilization. And here in the roadmap, we have already identified some standards that are related to sterilization. That is a huge factor that we need to consider in organ on chip, but the sterilization is only for healthcare products and medical devices. So we are still not sure, because we may use different materials, that these sterilization protocols will fit our own experiments, right? And yeah, I'm seeing you're nodding. Exactly.
Certainly.
It's difficult. But we already have a starting point, which is good. So we have already tested partially the protocols. We see they don't fit, they're not fitting our needs, but we can use part of these protocols and then develop the rest that is missing. So this is one effort that we are doing, and we need users to test this and to give their input on, well, but I have already done this with this protocol, why don't you test my own protocol? Let's see if that fits better than what no one knows how to do, or someone is trying to do, but it's not quite there yet. So the idea is these discussions, they help to harmonize these protocols with the inputs of different players. And end users in this part, they are really, really important. It may be that I know, in the materials part where you want to test the bending of the material, it may be not so important the end users. It may be more important that the manufacturers talk amongst themselves. But in this part of sterilization, for sure, it's super, super important.
Totally agree.
So, for example, how do you infuse your cells into the device? Is there like each user will do their own, and then the result will be the same at the end? Probably not, right? So there's a tweak. There are some things that need to be done in a certain way and with a certain sequence. And if that is in a protocol, in a guideline, that is a lot easier to then spread around. People test it, they give their input. It works, it doesn't, it needs this tweak, that tweak, it needs to change here, there, and whatnot. And in the end, the idea is to have one protocol that can fit everyone. Everyone knows that how they will infuse the cells into the devices, and expect that if they have differences in the final results is not because of the infusion, is because of something that happened inside the chamber. So something that is more related to the cells and not to the system.
Yeah. And how far do you think we are from reaching that point that we all do the same thing? Because for me, at least, you know, I'm using a particular device, and the decision we took in the lab was to buy a device from a company already. Okay, so we're not making our own. We're not doing
Probably already with the standard size and standard position.
Exactly. So yeah. So all these things, so already to minimize as much as possible, let's say, our user interference on the end product. But still, you know, we see that, I don't know, just by using different tips to pipette things into the device, the results are completely different. And if a bubble appears, well, in microfluidics, it's the end. So I totally agree with you, but for me, at least as a user that is getting expertise now, okay, it's still a very long path. I feel it at least, but perhaps you have a more realistic perspective on this.
It's a path, indeed. It's not very long. And I say it's not very long because we, as a community, we are working really hard to get things done. And a lot of people are engaged. Manufacturers are really engaged. There are several projects that, and as you know, more and more European funded projects, they ask for a standardization part, right? So I think it will be faster like that. What we know is that in 4 years, we will need to have standard procedures to assess and characterize metrological quantities in microfluidics, protocols to integrate elements and material characterization, general standards and guidelines to qualify quality for quality control and validation, and then a standard device to test several of these quantities. And we will have to have it. So it's going to happen. I think the effort, the push of both European Commission and, for example, the FDA on replacing animal models will fasten all this process. And what we are seeing is with these types of projects that are financed just for the metrology, so just to envision standards, in the end, it may be that the standard is not out because it's only 4 years, and you saw that the standards can be 5, can take 5 years to go out. We had the luck of, in one project, we were able to give input to 4 standards that came out within the frame of the time frame of the project, which is not common at all. But that only happened because a lot of people were involved, not only the partners in the project that were 15 in the other project, there are 19 in this one, but of the community. We had more than 30 stakeholders in the other project. We have more than 20 in this project now that is starting, but we will have much more while the project is going. So the involvement of the community is being more and more predominant, and that makes things go faster, a lot faster. Communication is key in this part. Like everyone is talking to everyone, trying to get this out, because also the manufacturers, they need this to sell their products. They will sell much more, so they are enforcing also the transformation, the use of standardization. Yeah.
Let's hope so. Very fast. Any other questions to Vânia? If I may ask you all to turn on your cameras so Paula can take us a photo of all of us on this first seminar, those that are still present here, and those that can, of course, turn on your cameras, those present here.
Let me just turn around and go to the other side.
Yes, so we can all take a photo, like we're back in COVID times.
I can speak. The speaker sits and the rest is not on the picture.
If no one else can turn on the camera, let's say hi, cheese to Paula on this first seminar.
Yeah, very, very nice.
Okay, excellent. Thank you.
Thank you so much.
Great. I will just share. Oops. My contact is easier if you contact me on LinkedIn. It's very convenient. If you have any questions, you don't know what to do next, but you have a nice idea, you want to contribute, but you don't know how. You want, okay. Or I don't know, you just want to know a bit more, don't hesitate in contacting me, because, or any of the people involved in the standardization of microphysiological systems, that we will most certainly discuss with you either your needs or your ideas, because we need ideas also, and we need arms to work, hands to work.
Definitely. So thank you so much once more, Vânia. Thank you. It was great. And if no one else has questions, we'll see you online on another virtual seminar next year. And I wish you all very nice Christmas and Happy New Year. Thank you so much. Thank you for this. Thank you. And bye-bye, everyone.
Bye-bye.
Thank you so much.
Thank you.
Bye.
Thank you so
you.
much, Vânia. Thank you. Bye-bye.